So today we're going to be talking about hairs and fibers and their significance to crime scenes, how to collect it, and what information we can pull from them. So we're going to start by talking about hair. Hair gets encountered as physical evidence in a wide variety of different crimes. Pretty much anywhere where there is some type of physical contact, you're going to have some hair left behind. You're unable to individualize a human hair to a single head or body, but it does allow you to note that there was physical contact between the victim and the suspect. You can be able, if you have enough hair, you can create a DNA profile and put someone at a crime scene and draw possible connections between a suspect and a victim. The more hair of someone that you find at a crime scene, the more likely they have been there more than at one time. When you're collecting hair, you do something called a paper envelope fold. I'm going to post a video on this in a little bit for you guys. You place the hair inside the paper envelope, which makes it seals it in. And then you place that paper envelope into a larger envelope, seal it with tape, initial, and give it a reference number as always. The morphology of hair. So what do we actually look for when we collect a sample of hair? Morphology is important to the examination of hair because hair has three separate layers: the cuticle, the cortex, and the medulla, and each tells us a different story about the hair itself. The cuticle is the outermost strand or outermost piece of the hair. It aids in the resistance to chemical decomposition. This is why most corpses who have been decomposed over a long period of time tend to still have a full head of hair. It's a scaly structure that covers the exterior of the hair. It's formed by overlapping scales that always point towards the tip of the hair, so away from the root, so towards the tips. It's formed from specialized cells that have hardened and flattened in the process of leaving the follicle itself. The scale pattern is not totally useful for individualizing human hairs, but it can help us determine a species of hair. Each species has a different individualizing or unique characteristic and pattern that helps you identify that hair as being from a specific species or not. The cortex is contained within the protective layer of the cuticle, so it's our next layer in. This is where the main body of the hair shaft comes from. It takes up about 70 to 80% of the hair shaft itself. And it derives major forensics importance due to the fact that it becomes embedded with the pigments of hair. So the more that you dye your hair, the more it will show in your cortex. If we were in class, you would be looking at samples of your own hair. And I do have samples of dyed hair. I can try to show you in your lab that you guys will get next week, but it's a little bit trickier to do. It's more fun when you can do it on your own hair. The color, shape, and distribution of the granules of pigment provide important points of comparison that tell how often someone dyes their hair, what colors have their hair been. Pretty much any color you dye your hair will leave behind traces inside your cortex. And the medulla, it's a collection of cells that creates a central canal that runs through the center of the hair. It's a predominant feature in most animal cells that occupies up to half of the hair's diameter, but not all hair has a medulla. It can be continuous, like you see right here. It can be intermittent, or it can be fragmented, or it can be completely absent. Typically, the lighter the hair, the less noticeable the medulla is. The root also gives pretty important pieces of information in a forensics way, because most times if hair is forced out of its root, you gain the surrounding cells with it that provides tools to be able to create a DNA fingerprint. If a hair is forcefully pulled out, you're likely to pull skin cells out with it. This is because hair grows in three separate stages. You have your anagen phase, which is your initial growth, and it can last up to 6 years. The root is attached to the follicle for continuous growth, which contains a bulb-like flame in appearance. So it'll be bulb-like. Think of like a light bulb, obviously not that big, but that shape attached to your hair. Then you have your catagen. This is when your hair continues to grow, but at a much slower rate. Roots typically will take on an elongated appearance. And then you have your telogen phase, which is the end of your growth phase, when your root takes on a club-like appearance to force its way out of the root itself, out of that follicle. Over 2 to 6 months after this hair grows phase has ended, the hair gets pushed out of the follicle, causing the hair to be naturally shed, so a new one can be grown in its place. When we're doing identification and comparison of hair, the primary purpose is to establish if the hair is first of human origin, and then second to determine whether the human hair that was retrieved at the crime scene compares to known or unknown suspects, or even our victim. So here we're looking at one family of animals. So we're looking at rodents. You have your red squirrel here, your porcupine, your Arctic ground squirrel, and your muskrat. So you can see that obviously our two squirrels look very different. They're all under 400 times magnification zoom, and your porcupine and your muskrat hair look different as well, but we're all from the same family. Microscopic comparison of hair uses a comparison microscope. It's an individual tool that allows the examiner to view the questioned and known hairs side by side to see whether or not they are or are not a match. The approach is considered subjective and highly dependent on the skills and the integrity of the analyst, as well as the morphology of hair is being examined. So we look for those, are the dye granules that are inside the cortex of the two samples of hair, are they the same? Because that is pretty rare for two people to dye their hair the exact same way. And it is subjective because to you, these two may not look like a match, even though I'm going to tell you that they are. So it is subjective. And you get analyzed, the hairs get analyzed by microscopic examination, chemical examination, and then DNA analysis. When you're examining, you're looking for these three main characteristics to be matching in the examination process. You're looking for color, the length, and the diameter. Are the colors roughly the same? Is the length of the medulla consistent? Is the diameter of the hair the same? Fibers, on the other hand, tell a little bit of a greater story. These may become important evidence in incidents that involve personal contact, like a homicide, an assault, or any sexual offenses. And there are two main types of fibers: natural fibers and manufactured fibers. Natural fibers are woolly. They're derived from an animal or a plant-based source. They come from nature. Animal fibers comprise most of the natural fibers that are counted at crime scenes. And natural fibers include anything from wool, it comes from sheep. You have cashmere or mohair, which comes from goat, furs from camels, llamas, and alpacas, and then even stuff like mink, rabbit, beaver, and muskrat. These are collected all in the same manner as hair, and they are examined the same exact way. The only difference is that we are looking for, is it natural? Is it manufactured? And then taking it into what is the chemical composition of the dye composed of the hair. Microscopic examination for hair and morphology characteristics happens to determine, again, what is the chemical composition? What is it a 60-40 blend, or is it a 100% blend? Things of that nature. The most common plant fiber is actually cotton. We use a wide variety of undyed white cotton fibers in our everyday lives, and they come in every form from clothing to other fabrics, which actually has made the evidentiary value of cotton absolutely meaningless, especially white cotton. But every company that uses cotton in their clothes has a different unique dye configuration. So Nike may use this particular blend of red dye, and they keep that as a log because obviously it's their, we'll call it their secret formula. But Adidas is going to have that same red shirt, but it's going to have a different dye composition and a different chemical composition of their dye. And again, that's on file with them. So when you find a red fiber and you're like, oh, let's see if it matches Nike or Adidas, you can pull up what their dye composition is for that specific shirt and that blend to see what manufacturer that shirt came from. This is what enhances the evidential significance to fibers found at crime scenes. Manufactured fibers began with the introduction of rayon in 1911. I don't know if anyone remembers what was going on in 1911, but it was around the time of World War II and women having to start going off into the workforce. So, sorry, I'm thinking nylon. I jumped ahead. So nylon in 1939, rayon was 1911, nylon was 1939. This is when it became really popular, when women had to start going off into the workforce, and they were expected to wear nylons under their skirts to give their legs a perfect clean line every time. This, at this point in time, increased and then replaced natural fibers in wardrobes. More people were wearing manufactured fibers. They were cheaper. They lasted longer. They were softer. All of those things. Materials were then processed and derived from wool or the cotton pulp. So they're not actually using the cotton. They're using the pulp to make a new fiber. They were able to pull cellulose from these plants and then chemically treat it to dissolve and create a desired fiber with a desired softness effect. Then manufactured fibers took it a step further to actually become synthetic fibers. These are manufactured and produced solely from chemicals. So they were developed when scientists developed a method of synthesizing long-chained molecules called polymers. This created a better way to make these synthetic fibers. And these synthetic fibers are actually what is cut into to make cotton blends. So polyester is a synthetic fiber, and most of our cotton blends involve some type of 60-40, 30-70 cotton polyester blend. Polymers is the basic chemical structure of all synthetic fibers. They resemble long repeating chains with each link representing a basic structure. And when joined together, they create one long strand that we call a polymer. And that's where polyester gets its name, because it's a long chain of ester molecules. The different polymers are what created the different synthetic fibers. When you're identifying and comparing fibers, the first thing you need to do, not only set it up microscopically how you would hair, but you want to start by identifying whether or not it is synthetic, manufactured, or natural. That's the most common starting place, because we actually have some synthetic fibers that are now also natural fibers. Silk is the most common example of this. Silk is considered a natural fiber, but it also can be made synthetically as well. Most of the fibers you collect from crime scenes will be unable to track in origin. So you want to determine if it's natural or manufactured, and then run that chemical composition on the color to known samples of fiber and known manufacturers that use specific color dyes. The techniques used in fiber examination. So there are a bunch of different things that we do to fibers other than microscopic examination, and one that we do is the visible light microspectrophotometer. Basically what it does is it uses visible light to break apart the different components of the fiber to determine what the dye composition may be. It's a convenient way for an analyst to compare colors of fiber through different spectral patterns. So when you say, oh, I have a red shirt, it's not really just made of red. There's a lot of other colors of dye in there to use to help design that red color. So the visible light microspectrophotometer helps break apart those spectrums to determine which pattern is used to make that color. We also can obtain a more detailed analysis of the fiber's dye composition through chromographs. And then we can analyze it through chemical composition to see if two fibers have the same composition of dye. We can do a bifringence. It's a physical property that shows the refraction patterns of crystals that are used in the makeup of manufactured fibers. And we can also do infrared absorption that works on manufactured fibers that absorb infrared light in a characteristic pattern depending on what their chemical composition is. The significance of fiber is evident. So once a fiber match has been identified, we have to answer a few questions. The first thing you need to do is, why is it significant to the crime scene? How does it tie to the crime scene? Why was this person wearing this shirt at the crime scene? You don't want to discount the importance of any one piece of evidence, including fibers as small as they may be, because it increases in significance if the analysts in the lab are able to draw a conclusion between the two samples. They're always collected and packaged in separate containers to avoid cross-contamination. The last thing you want to do is fold up your paper envelope with 3 pieces of blue fiber, 1 yellow, and 8 green fibers, because then you're going to cross-contaminate your fibers. So normally we would practice this. I am going to post a video of it in Classroom for you guys. So if you would like to practice folding a paper envelope, you may. But that's all I got for today.